In this video, we will discuss about how the cytoplasmic localization. So p53 is the protein that is usually found in the nucleus. So whenever there is AR stress, this paper makes a point that p53 will be localized to the cytoplasm. So let us understand their finding from the panel A. So DAPI will color or DAPI will stain the nucleus of the cells. So this is control. This is act as a control, and they have used p53 staining. So when there is no treatment or when there is no stress, the p53 will stain, and it is p53 will stain the nucleus, and the DAPI and the p53 can images are might seems resembles to each other. And after the cells are treated with tunamycin that we have discussed in the previous video, that it inhibits the protein glycosylation that leads to ER stress. We can see that the p53 staining can be seen in the nucleus, and also some of the staining can be seen in the cytoplasm, whereas there is no change in the DAPI staining. So there is no change in the nucleus. However, p53 localization is changed from just nucleus to nucleus and the cytoplasm. And same thing is observed when the cells are treated with thepsigargin. Thepsigargin introduced ER stress by disrupting the calcium homeostasis within the cells. And ER is one of the component or one of the contributor for maintaining the calcium homeostasis within the cells. So in this case also we can see that p53 is spread across the cells or in the cytoplasm, whereas in the control there is not much change in the DAPI. And when there is when cells are treated in a medium with no glucose, we can also see the p53 spread across the cytoplasm. And in control, that is in DAPI staining, there is no much change in the nucleus. So from this panel it seems like that p53 is localized in the cytoplasm whenever there is a ER stress. And same thing they have done with the another cell lines, that is HT1080 cell lines. In this case also they can see that the p53 staining in the cytoplasm whenever they have introduced the ER stress through this tunamycin or thepsigargin. Then in order to confirm this thing, they did the Western blot of the subcellular fraction of the cells that are treated with thepsigargin. So here the first three bands are the whole cell extract that shows the p53, uh, and this that shows the p53 uh, staining in the panel A, and these cells are treated with tunamycin. So we can see that with time the p53 amount seems to be reduced, however it is present within the whole cell extract. And when we check this thing in the cytoplasm, we can see that there is increase in the amount of p53 with increase in time. And we check this content in the nucleus, we can see that there is a decrease in p53 with time. So this thing supports the observation that observations from the panel A and panel B. And same thing they did it with the cells that are treated with thepsigargin, and similar observation are found that in cytoplasm with time there is a localization of the p53, and in nucleus there is a delocalization of p53. And in order to confirm the quality of this quality of this subcellular fractionation, they did the check. They used two different kind of antibodies such as ERG1, that is mainly represent or mainly present in the nucleus fraction, and alpha tubinil, which is mainly present in the cytoplasm. So subcellular quality is also good in this experiment. Then they did another experiment, that is northern blot. Northern blot we do, instead of using in northern blot we use mRNA instead of using the protein. So, and they did the experiment on the lithium bromide gel, and they can find that, and they did this experiment on the whole cell extract. So they find that uh, there is no change, there is not much change in the mRNA mRNA content of p53, and this thing is validated with this 28S RNA and 18S RNA. So this shows that ER stress does not affect the mRNA stability. However, it might leads to localization of p53. In our previous video, we have seen that ER stress also affect p53 stabilization. However, in this figure, the researchers is trying to point, trying to make a point that ER stress causes p53 localization. And then they did another pulse. Then they did another experiment to prove this point, or to uh, make this point to, we can say, then the researchers did another experiment to further substantiate their point, that is pulse chase experiment. So what they did is they treat the p53 cells with the radioactive cysteine or methionine, and then transfer them into the normal medium, and at different intervals of time they collected the samples and then measured uh, the radioactivity through the phospho-radiographer, or you can say phospho measurement, something like that the instrument name is. And so with time they measure the radioactivity, and they can see that in this panel, at with time the amount of p53 reduces, amount of p53 decreases, or we can say that is been compared with the help of this radioactivity, and they can see that amount of p53 reduces in the thepsigargin treated cells is more compared to the no treatment. Or we can say amount of radioactivity, percentage of radioactivity decreases in the no treatment is at a slower rate compared to the amount of radioactivity decreases in the thepsigargin treated cells. So this shows that the this gives a point, or this shows a indication that p53 proteins in the thepsigargin introduce cells, or thepsigargin affected cells, or we can say in the ER stress cells is less stabilized compared to the wild type cells. So from this they made a point that ER stress might destabilize 53 protein, and then they did the experiment that has been discussed in the previous video. So if you need to understand this story in details, or if you need to understand this study or research in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.